All right, class, can y'all hear me? I know,
Yes.
okay, great. I know most of y'all's mics are muted and that's fine. So first, let me say thank y'all for being here. I really appreciate this and sorry for having to change this last minute. But just keep in mind that all the rest of the Zoom meetings will be on the dates listed in the syllabus. This is the only one that'll be on a Thursday. But what the purpose of this Zoom session is it's kind of like the exam review. I know the exam is not until September 21st, which is over 2 weeks away, but this is the last time we'll discuss chapter 2. So we finished chapter 2 and tomorrow is your chapter 2 quiz. So keep that in mind. We'll talk a little bit about what you'll see on that quiz. And then a lot about what you'll see on the first exam. So on Blackboard, this is posted. This is your exam review. You can see 50 questions. It's about 20 questions from chapter 2 and about 30 questions from chapter 3. So the next Zoom session is on September 18th and we'll focus only on chapter 3. So today is all about chapter 2. What I'm going to do first is I'm going to go over each of these bullet points and then I'm going to pull up the PowerPoint and kind of talk a little bit more about more information, just kind of the really tricky parts that you may have encountered while learning this information. And then I will open it up to y'all and y'all can type in the chat box on if there's any other questions you have or anything you want me to go over further. Okay, so let's kind of just get started looking at our review here. So one thing that I added to my document here that you don't have on your document is this first bullet point, energy conversions and the first law of thermodynamics. So that's kind of the first thing. Hold on a second. And that comes from just this slide right here. So this slide here, we kind of talked about the different types of energy. So what you have to know is just what's the main energy conversion from putting food into our body and how we use that for exercise or we use that for muscle contraction. So knowing just that energy conversion from food to using it for energy. And then the first law of thermodynamics. So look back into the lectures. I'm not going to say it again here, but in that lecture, we talked about what the basic definition of that is and how we can apply that definition to the energy systems. Next one is looking at, hold on. Next one is function of enzymes and coenzymes. We probably already know what this is here. We've learned about a lot of enzymes in chapter 2, so in the phosphagen system and in glycolysis. What you have to know is the primary enzymes, which are listed right here. So kind of the main enzymes that you have to be familiar with is creatine kinase, and we'll talk about that role in just a little bit. Hexokinase, lactate dehydrogenase, phosphorylase, and phosphofructokinase. So those are the main enzymes you have to know. If you see any other enzymes in this lecture, you don't have to know them for the exam. And then coenzymes, we briefly mentioned what the role of them were. We referred to coenzymes. I think our example was B vitamins are coenzymes for a lot of enzymes in glycolysis and in our energy metabolism. So without B enzymes, those systems or those enzymes don't function as well. This one right here is just terminology. So it's just knowing those definitions. So it's just definition questions, being able to identify the difference between glycogenesis and glycogenolysis and then gluconeogenesis. I think gluconeogenesis is actually talked about in chapter 3. So if you can't find that in chapter 2, just wait till chapter 3. I think that's where this one right here is from. And then next, formation and hydrolysis of ATP, the depletion and accumulation hypothesis. So that's going to be the first topic that I'm going to review because it's one of the most important. So this one and then glycolysis I'll go over next. So let me switch back to the PowerPoint. Okay, and I kind of just want to start right here. So just kind of talking about that depletion hypothesis and accumulation hypothesis. This slide here refers to the accumulation and how ATP is formed and how it's hydrolyzed. This question should have been answered on your assignment. I think the last question, that critical thinking question. We'll get into why we can't sprint indefinitely, but it all starts with this accumulation hypothesis. So let's talk, why is that? So we know ATP, back here you can see the formula, which if you refer to this, you can see what it's made of. And then this slide here, we looked at endergonic and exergonic, or anabolic and catabolic reactions. We talked about how we can form ATP, what does it take to do that, and then also what happens when it's broken down. And that's what this slide here is. So we know that when we exercise short term, we're pulling from our ATP stores in our muscle. We don't have to start a system if we're exercising for 6 seconds or less. So think of like a 6-second sprint or a bout of cycling or a bout of resistance training. We're just pulling from our muscle stores of ATP. They're those cellular stores. But when we pull from them, when we pull that ATP to use it, we have to break it down in order to release that energy, which you see right here. So when that energy is released, that's great. We have energy for exercise, but we also have the accumulation of phosphagen and hydrogen ions. When they accumulate in small amounts, that won't cause acidity. So just small amounts is normal to be circulating in our blood. But it's when we're breaking down ATP rapidly that phosphagen and hydrogen accumulate in high amounts. So this is without rest. If we continuously exercise really high intensities for long periods of time, we have that increase in acidity due to accumulation of phosphagen and hydrogen. That's the accumulation hypothesis. That's one reason why we can't sprint indefinitely, is because our muscles become acidic. You've probably felt it, that burning sensation that sometimes makes you nauseous. That's the accumulation hypothesis. So that's kind of the ATP hydrolysis part. But now let's flip to this slide here and talk about, okay, well, what happens if we continue to exercise past that 6-minute mark, now into the 10 to 30-second time frame, still of really high intensity? What does our body have to turn to next? So in order to make more ATP, because we already know it depletes very rapidly, what we have to do is make more of it. And how do we do that? We turn to creatine kinase. So creatine kinase is the primary enzyme in the ATP-PC system, where it's a catalyst. It's a facilitator for this reaction right here. So phosphocreatine, creatine and phosphate, it is held together by a high energy bond, which you see is that wavy line right here. So that high energy bond is going to be broken. And when it's broken, that's going to release energy. And then we can take that phosphate and we can donate it to ADP. And that's how we make more ATP. So that's how we have more energy. So this process is mostly working during that high intensity time frame from about 30 seconds or so. So it's kind of the role of creatine kinase, how we make more ATP. But we also know that ATP and creatine phosphate, those are going to deplete very rapidly. So this picture here, this graph is from 14 seconds. This depends on the person. It depends on how much ATP and creatine phosphate is stored in that individual's muscles. So this, we can extend this up to about that 30-second mark, 20 to 30 seconds or so. So just remember, this is all kind of based on the individual. But for the most part, those depletions occur very quickly. So within about 14 seconds, we have almost 100%. We see it depends on which fuel source we're looking at, ATP or creatine phosphate. We deplete that. And if we deplete those fuels, we don't have as much energy, or we don't have any energy to continue at that high intensity. So what do? This right here says we have to take an aerobic recovery period. So an aerobic recovery period is just like walking for a second. So let's say you did a 20-second sprint. Now walking for 10 seconds, 20 seconds or so, that will help to replenish those stores. And then you can go into another bout of high intensity exercise. So that depletion hypothesis was also, or should have been, in your answer for number 5. So discussing how we deplete those fuel sources, we kind of hit fatigue. Or when we have accumulation of acidity, we also hit fatigue. So that's where, or a reason why we can't sprint forever. Depletion of fuel sources, accumulation of acidity. Okay, so let's flip back to, so that was kind of the main topic that you have to grasp. So that's one of the most important topics in chapter 2. Let me flip back to the review and we'll talk about some other important concepts. Okay, duration of each energy system. This one's pretty simple. Again, it's just like how long does the phosphagen system last? We just went over it, up to 20 to 30 seconds of high intensity exercise. Glycolysis, when does that system become the primary energy source? That's going to be from the 30-second to the 2 to 3 minute mark. And then when does aerobic metabolism become the primary energy system? After that 3-minute mark of low intensity exercise. All right, now that gets us into glycolysis. So that's going to be the next topic I go over, and then I'll probably turn it over to y'all for questions that you may have. So glycolysis, you can see here, we have to know what is being produced in glycolysis. So ATP and then NADH. So those are really the only byproducts, but we're going to talk about how many of them are produced, what happens to them, and then also what determines fast versus slow glycolysis and what happens to pyruvate. That was also a question on your assignment that you probably answered correctly. So let's get into glycolysis next. Flip the screen back over. All right, so we're in the glycolytic system now. So the way I want you to think about this is you've been exercising for longer than that 30-second mark, and it's still a pretty high intensity. So it's not slow, it's not considered easy, it's considered moderate to high intensity, and you're going to continue that exercise for about 2 minutes or so. So think of something that you can continue for about 2 minutes, but after that 2-minute mark, you're probably going to be pretty tired. You probably need to rest or lower your intensity. So that's how you want to think of glycolysis. So I know when we look at this chart right here, just like I explained in the videos, there's a lot going on, and it's pretty daunting. But when we break it down, it makes it easier to understand. So let's just kind of start again at the top. So 1 molecule of glucose, that's what we're talking about, just one. Think of all the billions and trillions of glucose molecules that we consume on a day-to-day basis. So this is 1 glucose molecule that enters into our muscle cell. So when it enters into the muscle cell, first, it can come from 2 different places. So that glucose can come from our bloodstream. If it came from the bloodstream, it means that we had recently consumed food or a sports drink or a sugary beverage. So we've recently put something in our body that contains carbohydrates. It contains sugar. So we take that glucose molecule, and we can see it's a 6-carbon molecule. And this enzyme here, hexokinase, is the enzyme that starts glycolysis if we're using glucose. We know here already that we have lost an ATP. We lost a phosphate, so that was lost. We're kind of down. We're down a energy molecule. We lost one. So we invested it though. We invested it knowing that we're going to get it back. And then we already talked about how we go through these different steps here. We talked about this phosphorylation phase. Phosphorylation is the addition of a phosphate group. So when we lost that phosphate, where did it go? It went right here to glucose 6-phosphate. And then it transfers into fructose 6-phosphate, and then phosphofructokinase, PFK, is the rate limiting enzyme. if you remember, that rate limiting enzyme says, or basically it determines how quickly the entire system works. So it determines the rate at which all those reactions occur. We also lost another phosphate, we lost another ATP. So we're down 2 now. And that phosphate was then transferred here. We see fructose 6 phosphate, and then we see fructose 1,6 biphosphate. So now we have 2, that's where that phosphate went. So that's what happens. We lose 2 ATP in the investment phase if we started with our blood sugar. But what about if we started with muscle glycogen? A little different. So when would this occur? So let me give you an example. This morning at 6 a.m., I went for a run without consuming any food, without drinking any sugary sports drinks. So my blood glucose was low. I had just woken up from an overnight fast. I haven't consumed anything since dinner. So my blood sugar is low, meaning I can't use blood glucose for energy. I have to turn to my muscle glycogen. So I turn to the muscle glycogen, I can pull that glycogen out of the muscle, and the enzyme phosphorylase is the enzyme that's going to break glycogen and turn that into glucose 1-phosphate. But this reaction right here, the phosphorylase already phosphorylated muscle glycogen, so we don't have to lose or invest in ATP. We don't lose anything in this first conversion, and then everything's the same. So we see here loss of 1 ATP using glycogen. So from this standpoint, it kind of looks like glycogen is going to be the better fuel source to produce more ATP in the long run. We then move down here, and we're in that next phase. I know there's a lot going on here. Again, you don't have to worry about ADALase. That's just the enzyme that turned fructose 6-phosphate into fructose 1,6 biphosphate, just kind of extra information. And then we see here that the reaction splits. So it's going to split, and every time there's a splitting in the cell or splitting of the reaction, it means the cell is becoming more unstable. And if the cell is becoming more unstable, it's more likely to give off its energy. And so that's the purpose of going through all these different reactions, is we want the cell to just give off energy so we have more ATP. So kind of what do we want to focus on here? So if you look, it's identical. So each side is identical. And what's happening here is we're either losing a phosphate, we're losing hydrogens and electrons, or we're just transferring phosphates all the way down the chain. So here, let's talk about that NADH production. And this word right here is what NAD stands for. And then you add the H, and that's hydrogen. But nobody wants to say this word, so that's why we just say NADH. All right, so NADH, or during this reaction right here, we're going to lose a hydrogen and electron and its associated electrons, which is picked up by the NAD, and now we have NADH. So NADH, we haven't focused a lot on that molecule yet, but it can be transferred or sent to the electron transport chain. When does that happen? That will only happen if there's enough oxygen present in the cell. You probably know, I just didn't mention it, is that glycolysis and the phosphagen system are anaerobic, means they don't need oxygen in the cell. It doesn't mean we're not breathing oxygen to fuel that exercise. It just means there's not oxygen in the cell, and that oxygen is not needed for those reactions to occur. But the electron transport chain, it does rely on oxygen. So if there's enough oxygen present in the cell, then NADH can be sent over to the electron transport chain, and that's where it will produce more energy and produce more ATP. But if there's not enough oxygen in the cell at this moment, meaning we're exercising at a really high intensity, then NADH cannot be sent over to the ETC, but it can be sent down here to the very bottom of this reaction, which we'll talk about later. But okay, so as we keep going through here, we now see that we have produced an ATP. So we've produced 2 on either side here, meaning they just lost a phosphate, or we gathered a phosphate. So there was a phosphate that was split off the reaction, and it was picked up by ADP, which turned it into ATP. And then we see here, we go from 3-phosphoglycerate to 2-phosphoglycerate to phosphoglynyl pyruvate, and then all the way down to this reaction right here where we produce another ATP. So again, we're just making the cell more unstable by forcing it to undergo reactions, allowing it to give off its energy. So now we have 4 ATP that were produced. End result of glycolysis is pyruvate. Let's talk about what happens to pyruvate. So you wrote about this on your assignment when it asked kind of what's the fate of pyruvate, what happens to it. So we have 2 different directions, and that's going to again depend on the rate of energy demand. So let's discuss that. So pyruvate kinase, that's what turned that last ion into pyruvate, the enzyme. And then we see here we have 2 directions that we can go. So let's first talk about fast glycolysis. Lactate dehydrogenase is the enzyme that turns pyruvate into lactate if there is not enough oxygen present in the cell and there's a really high rate of energy demand. So what I want you to think about is you're exercising at a really high intensity and your muscles aren't using oxygen, but we're still producing energy. That pyruvate then will turn into lactate, and the process keeps repeating itself. So glycolysis will keep repeating itself, producing more lactate. Lactate is not the main cause of fatigue, but it is associated with fatigue. And we'll talk more about that concept in chapter 3. But how do we turn pyruvate into lactate? Well, that hydrogen there, that NADH that we produced up at the very top, it's sent down to the bottom here, where then pyruvate can accept that hydrogen and it can turn it into lactate or turn pyruvate into lactate. So that's actually one way to decrease hydrogen, so decrease acidity in one way, but then when we produce lactate, that causes fatigue in a different way. We can send lactate to the Cori cycle and we can produce more glucose, but that's a chapter 3 concept that we'll discuss later. So that's one way, but think about it, it's all due to the rate of energy demand. If I'm exercising at a really high intensity, I need ATP very quickly. So I have to keep going through glycolysis because glycolysis produces ATP very fast. But let's say I slow down my intensity. So I slow down, instead of sprinting, I start to slow my intensity into a light jog. Now I go into slow glycolysis. So pyruvate is sent to the Krebs cycle. This would occur if the rate of energy demand is lower. So if I'm doing a light jog, I don't need ATP as quickly, but I'm going to need a lot more of it to sustain that light jog for, let's say, 30 minutes. So that's where I send it to the Krebs cycle, and we talk more about that process in chapter 3, like what happens to pyruvate after that. But just to kind of sum up glycolysis, we know that 2 ATPs were produced if we started with blood sugar, started with blood glucose. 3 were produced if we started with glycogen. We also already talked about how hydrogen ions can be accepted by NAD to make more NADH. But if aerobic metabolism cannot accept hydrogen because there's not enough oxygen present in the cell, the intensity of our exercise is too high, pyruvate will then accept the hydrogen and it will become lactate. That's what you see down here. These 2 reactions just kind of show that. And then it just kind of shows it again. But yeah, so that's kind of all I wanted to discuss. Let me switch back to, this guy right here. So that was glycolysis. Training adaptations to glycolysis, that's going to be a big question on the exam. There was a question on your assignment about the training adaptations to ATP-PC system, and a lot of students actually missed that one. So I did want to spend just a few more minutes discussing those training adaptations, and then I'll be finished, and then I'll turn it over to you guys. Okay, so coming back here. All right, so let's get into chapter 4. So this slide right here was the answer to your question on your assignment about, I think it was number 2. What are those training adaptations to this system? I probably worded the question weird where students didn't really gather what I actually wanted, but this is what I wanted. So when we train the ATP system, that system is going to undergo adaptations to make it more efficient. So our body uses that system better. So what's going to happen is increased activity of major enzymes. We really only talked about one primary enzyme, which is creatine kinase. So one adaptation is creatine kinase works faster. And if creatine kinase works faster, we're going to have faster ATP production due to the hydrolysis of phosphocreatine, donating phosphate to ADP, making more ATP. So basically, if I train my ATP system, meaning I do a lot of sprints, I do a lot of plyometrics, high intensity weightlifting, that system is going to become more efficient and creatine kinase is going to work better. Second adaptation is elevated resting concentrations of ATP and creatine phosphate. So I have things or little statements from studies right here that just talk about how creatine phosphate and ATP can increase in concentration. It showed with resistance training. We see that we did see a positive relationship with doing high intensity resistance training and increasing our stores. We saw here that the sprint training, the levels actually remained untrained, but performance did increase. But this probably had to do with the sprints were not long enough in duration. They may have been too short. And then endurance training, we didn't see any significant changes to concentrations, but more recent studies since 2002 have shown increases. So just more ATP, more creatine phosphate means you can exercise at high intensities for longer before fatigue hits. So that was on your assignment. This right here will be on your exam. So just understanding if I train my glycolytic system, if I do a lot of exercise at high intensities for 2 to 3 minutes without stopping at that really high intensity, what's going to happen? We're going to see changes in these enzymes right here. So these enzymes will be more efficient, they'll work faster, which may result in more ATP production. We'll see increased intramuscular glycogen stores. So our glycogen stores may build up inside the muscle, meaning that we have more energy that we can pull from and we can exercise for longer because we have more fuel. And then last was improved buffering capacity, meaning that we can clear away acidity. We can clear away those hydrogen ions. So when we clear away hydrogen ions, again, it means we can exercise at higher intensities for a longer period of time before acidity starts to take over, burn our muscles and kind of make us feel sick. And then this slide here just kind of talks more about creatine phosphate and those buffering systems. But yeah, I think the last question on your exam review was about the interactions between anaerobic and aerobic metabolism. So this isn't understanding like a definite concept, it's just kind of understanding it as a whole. So kind of starting right here, this just showed like a 3-second sprint. There's still some aerobic metabolism contributing to that ATP production. And then these statements here say like the longer the sprint, the more we have to rely on more glycolytic and aerobic energy system. Repeat sprints use more aerobic metabolism than the first bout. And this just kind of sums everything up with those interactions. Just know from those last few slides right there is it's never like 100% the phosphagen system or 100% glycolysis. It's going to be a mixture of both. One energy system is never like shut off. They're always working. They're always contributing to energy. They're always producing some ATP for us to use, but one system will always be the primary contributor. One system will always be giving you the most amount of energy. And what does that depend on? It depends on number 1, the intensity, and number 2, the duration. So yeah, that kind of sums up what I wanted to go over with y'all, just to make sure we heard the most important information just one more time. Give me a second and I will open up the chat box and y'all can ask questions or we can go over something else. So kind of let me figure out how to do this. Okay. All right, I can see the chat box now. Does anyone have any questions over anything you want me to talk about, go over more? I'll just wait a second if you guys want to think of anything. Y'all can still see me, right? Can you see me? Yes? Specific enzymes. Yes, Easton, let's do that. Are you talking about the ones in glycolysis? Just kind of their main function?
Could we go over, okay, I'm just gonna talk.
Yeah,
so, okay, so could we go over the enzymes that was like that second bullet point? Like that had like the hexokinase and everything?
Yeah, just wanting to know more information about each one of those.
Yeah, like the
Okay.
specific function of those. That's like the
Yeah.
only thing I'm tripped up on.
Okay, cool. Let's do that. So starting, okay. Let me get to my screen share again. Okay. All right, so most important enzymes. The order that, hold on. All right, so we've got creatine kinase, hexokinase, lactate glucose. Okay. All right, let's start with hexokinase. So what you need to know about hexokinase is it is the primary, it's the enzyme that basically starts glycolysis. It's the enzyme that takes blood glucose, a 6-carbon molecule, and turns it into glucose 6-phosphate. Hexokinase is also where the phosphorylation process occurs, and we can see that here, that blood glucose, that 6-carbon molecule, we have to add that phosphate, and that's why we lost an ATP, because we needed another phosphate to make that glucose 6-phosphate. So hexokinase was the enzyme that facilitated the reaction between the 6-carbon molecule to the 6-carbon phosphate molecule. So it's just the enzyme that turned blood glucose into glucose 6-phosphate, and it started glycolysis. Okay. We also lost an ATP right there, and that's also where the phosphorylation occurred. Phosphorylase does the same thing, but to muscle glycogen. So it takes that muscle glycogen stored inside of our muscle tissue, whatever active muscle that's working, and phosphorylase, it's actually called glycogen phosphorylase, but this here just says phosphorylase, but just know it works with glycogen. Okay. So it takes glycogen and it also turns it into glucose 1-phosphate. Phosphorylase, we didn't have to lose an ATP in that conversion or in that reaction, because phosphorylase already added that phosphate, which we see right there. Hexokinase couldn't do that. We had to lose an ATP to get a phosphate. But muscle glycogen, phosphorylase is the enzyme. It converted it to glucose 1-phosphate. Phosphorylase just added that phosphate without us having to lose ATP.
Okay.
Want me to keep going with PFK and LDH? Yeah, sure. Okay. Phosphofructokinase, right here, rate-limiting enzyme. We also lose an ATP right here. So phosphorylation also occurs in this step where we take a phosphate and add it here. PFK, just really what you have to know, it's the rate-limiting enzyme of glycolysis. It determines how quickly this entire system works. Everybody's PFK works differently. I think I talked about this in the video, that everyone has a different metabolism, meaning some people's have a really fast metabolism, some slower. Your glycolytic metabolism is determined by PFK, the rate at which it works, how efficient it is, and how quickly it can cause all those reactions to occur.
I mean, just a side note, would you also say that's determined by like if you're like an ectomorph or an endomorph or a mesomorph?
Yeah, sure.
Okay.
What Easton's referring to is body type. So there's different body types, very thin, like an ectomorph. A mesomorph is muscular, and an endomorph is more rounder body type. And yeah, body type is also kind of determined by how quickly energy systems work, but then also genetics. So.
And so, if you don't mind me asking, how would you phrase this on the exam if you are gonna give us that information? Not like word for word, but like how might you ask it?
Right. So I may ask this like, just for this enzyme or just all the enzymes?
Just like if you were just gonna ask a question over it, like,
Um,
you know,
yeah, so a lot of this exam is actually very straightforward. I think this is the easiest exam because it's
okay,
all like definitions or like, what is this? So a question that you may see may be like, what is the rate limiting enzyme in glycolysis? And I'll have a bunch of enzymes listed.
JFK.
So that's like a really easy question, a way to answer it. Another could be like, listing like this enzyme, the role of this enzyme is this and this and this. What enzyme is it? So this exam I think is very straightforward with like asking like how many ATP are produced in glycolysis, or what is this enzyme, or kind of stuff like that. So I think it'll be pretty straightforward.
And so, like another general, I don't mean to be hogging all the questions
That's
right
okay.
now. All I'm hearing is crickets. So, like, what would be the most missed concept, would you say, like on past exams in regards of this first one? Like, which one, what's a point do you think we really need to hammer home before?
It's understanding like the big picture. It's
Okay.
understanding like it's like putting it all together. Like if I give you like, I'm doing this exercise, like this is the exercise I'm doing, and this is how long my exercise is, and this is how intense it is. What's the primary energy system that my body's using? So it's stuff like that. It's like not just answering the what is this or whatever. It's putting it all together. So like if I give you an exercise, if I give you an activity, a duration and intensity, it's knowing like, oh, they're using the glycolytic system based on what the workout looks like. So kind of stuff like that.
Okay, so like if you're doing short interval sprints for 20, 30 seconds, you're going to be using the creatine phosphate system. And this is what it's doing to break down like ATP at this accelerated rate. So you can do that for a short duration.
Yes.
Then you need to take a break so it can like restore the ATP stores and everything before like all that buildup happens. And then you can't do it anymore. Okay.
And another big question, this is like the most missed question that I always have, but I still haven't changed the question, hoping that one day students will answer it correctly. It's basically talking about like, if I'm doing an exercise similar to what I just said, what fuel source is going to be depleted the most? Like what fuel source will I deplete if I'm either fasted and doing this exercise? What fuel source will I lose or be depleted? Or if I've eaten food, so if I'm in a fed state, if I've consumed food and I am doing this type of exercise, what fuel source will be depleted? So it's just like that question I said earlier, but now talking about what fuel source will I be depleted the fastest, dependent on my current state and during exercise.
Okay, so like when you said you came from a fasted state from sleeping and you went for like a 6 a.m. run and you didn't consume anything, so you're not going to have any glucose to immediately spare, or you're going to be low on it, so you're going to go into those glycogen stores and start pulling from that. And so you're technically like more efficient from ATP, right?
Sorry, what'd you say? More?
You're technically more efficient in ATP since it's going from the glycogen, right?
Yes,
okay.
Yes, I can produce slightly more ATP in that way. And then once we get into chapter 3, we're combining that too, because that's when we talk about aerobic metabolism. So then when we get into chapter 3, you'll have to know, like, when is aerobic metabolism used? What fuel source will be depleted there? And stuff like that. So we got to grasp chapter 2 because we're just going to build on top of that for the other two, or with the other chapter.
Okay. And so, okay, last question. Okay, so when we figure out chapter 3, And then will we have like combination questions, or are they gonna be like strictly separate?
You may have a few combination questions, just again, the big picture. Like
Okay,
if
yeah.
I started at a 20-second sprint and I went and I kept running for 2 hours and then or sprinted in between there, just having to know, okay, so this is what they're doing. This fuel source is going to be most depleted. This is the primary energy system. This is what their body is using for energy, kind of stuff like that. So a few questions where you've got to understand just the big picture of how all the energy systems work and produce energy and stuff like that.
Okay, cool. Thank you.
Yes. Anybody else have a question? If you want to type it in or speak, whatever. I'll give y'all a second to think about it. There's a lot of people on the call, which is really cool. That's great. Let me see, where's my chat box? There's my chat box. I'll give it a second. If anyone wants to think, go through the slides, go through stuff like that. Yeah.
Okay, I have a question, but I don't
Yes.
know what typing it. So in the book, it says that at the end of glycolysis, pyruvic acid turns into lactate. Is there like a difference? Because I know pyruvic is the conjugate base of it. Or
no, so pyruvic acid and pyruvate are similar. When we add acid to the end of it, it means it's accumulated. So like if like pyruvate is one single pyruvate molecule, pyruvic acid is a lot of pyruvate molecules, just like the difference between lactate and lactic acid. So lactate is just one lactate. Lactic acid is a lot of them accumulated. And when they accumulate, pyruvate necessarily doesn't cause any issues, but it does turn into lactate, which produces lactic acid. I just didn't really refer to it that way, but thanks for the question because that is
okay.
helpful.
That makes a lot more sense. So I was like, what the heck?
Yeah, sorry. That's what it is. Anyone else? I'll give y'all a second just to think about it. All right, well, one more thing I'll just briefly mention is your quiz 2 review. So your quiz is tomorrow. You probably already know this, so keep it in mind for every single quiz. It opens at like 7 or 8 a.m. in the morning, I think 7, and closes at midnight. So you have the entire day to sit and take that quiz. Once you sit down and take it, I think the time frame is 15 minutes, which is not very long, but there's only 10 questions. And there's a lot of fill in the blank. Because actually if we were to have this class in person, every question would be fill in the blank, which is pretty tricky and you don't get any notes. But with this online class, you do. So it makes it easier to take these. So this here is a review. They'll all look like this. They're kind of broad, kind of vague. But types of reactions for your quiz, you just kind of can know that that is anabolic, catabolic. So just knowing the difference between those. Anabolic, catabolic reactions, the role of enzymes, what's their main purpose, what do they do, how do they help reactions occur. Mass action effect, I went over that in pretty good detail in the video lectures. Lock and key concept, also went over, that's just like enzymes and binding to substrates and stuff like that. Important enzymes, we just went over that one. Again, it's just kind of very vague. It's kind of just like main points of the lecture, kind of the most important topics that we discussed. Yeah, so, oh, and this one right here, don't get confused, net production of ATP versus gross production of ATP. Gross production is like, I produced 4 ATP in glycolysis. That was how many I produced. But the net production, we have to take into account what we lost. So we lost 2. So that means that our total production, our net production is 2, but our gross production, we actually did produce 4, but that doesn't take into account what we lost. So that's something else that you do have to keep in mind as well, net versus gross production of ATP in glycolysis. But yeah, is there anything else? I'll give y'all just a few more minutes if you want to reach out and say anything. And then we can end the session if anyone has anything. Okay, functions of protein. Which functions of, oh, the role, like the role of it, the substrate?
Yes.
Okay, great. Let's go look at that real quick.
And then, like, when would we actually use it for energy, or is it just for, like, maintaining and growth?
Yeah, let's talk about that because I briefly mentioned it. Okay, let me go back to my screen. Okay. Okay, you also wrote about this on your assignment, just briefly summarizing the role of each of these sources. So protein, we know protein is, the main role of protein is basically muscle rebuilding or muscle building, so like hypertrophy, growth of muscles. It also helps to transport nutrients. It helps with like temperature and hormone regulation. It transports hormones throughout the body. So its main role for the purposes of this class is going to be muscle rebuilding. So think about after resistance training or after any exercise, really, our muscles may have small micro tears in them. They may just be, shredded. Something may have occurred during the exercise. And if that's the case, protein will help to repair that muscle that we damaged. And then also, if we have excess protein, it will help to build up the muscle tissue to make it larger. So that's the main role. But when is it used for exercise? We see here only a small amount of protein is metabolized to provide energy. I think it's in chapter 3 where we're going to talk about this in a great amount of detail because protein is mainly used in aerobic metabolism. Protein is not used in glycolysis. So no protein, no amino acids can enter into the glycolytic pathway. But when we move into aerobic metabolism, which occurs in a different part of the cell, amino acids can actually enter into it. And amino acids can enter into this part of the cell, meaning we can use amino acids for energy. This is something that we actually don't want to do. If we're using protein for energy, it means we may actually be pulling from our muscle tissue and wasting our muscle away. So like losing it, which we don't want. But protein, again, a very small amount of it. And I think in chapter 3, I give you like percentages of protein use during like a 2-hour run versus like a 5-hour run. So we'll talk in more detail about the role of protein and how it can enter into aerobic metabolism and what it does. But for the purposes of this class, it's mainly to help repair muscle tissue after damage.
Thank you.
Yes. Okay, anybody else? I'll give y'all just a few more minutes if you want to come up with an answer. Or come up with a question, not an answer. Answer to the first question. What first question are you talking about?
Whichever one pops up first on the quiz tomorrow, you know, is it like A, B, C, D? You know, I have to ask that question. It's a
Yeah, I
classic
know.
one.
So what's actually neat on my end is everybody's questions are going to be different. So it's going to be a different answer for everybody.
Okay, so in that case, what's the first specific answer to my question? I mean, everyone else can ask the question for themselves, but like, what's the answer for my question?
What are you talking about?
I have my notes out. I'll write it. Like, I promise I won't tell anyone.
No,
Come on.
I have no idea what your first question is going to be, but it'll be easy, I promise.
Okay, I'll take your word for it.
It'll be fine.
All right.
All right, last call. Anybody else need any additional help, questions? You can always email me, and you can always request to Zoom with me individually if you would like to do that. So whatever you would like. But if there's no more questions, then we can end the session. Oh, wait. When will we get our assignment grades back? Okay, so I've already graded some of them. I'm going to spend the day grading some more of them, but probably not till the end of the weekend. But I'm going to try to get to everybody's like today and tomorrow. So today and tomorrow, I'm going to try to grade them all. But if not, I promise you will have your grade back by Sunday. Okay. All right. Well, if there's no more questions, I'll end the session. And remember, again, shoot me an email, ask to Zoom. And yeah, I hope this was helpful. Just more information, just kind of helping you guys out, doing what I can on my part since this is such a crazy time. But yeah, so have a lovely weekend. Take your quiz tomorrow, and I'll see everybody in the next Zoom session. Okay? All right, bye.